Okay, so in the previous video lecture, we have ended our previous video lecture with a question that how many clones we have to study or we have to screen to find out our gene of interest in a particular genomic library. Okay, so I want to mention another question that during the construction of genomic library, how many clones of cell you required to complete your entire genomic library. Okay, so we will going to discuss about both the two questions, that how many genomic clones we require to complete our entire genomic library formation, and similarly how many recombinant clones we have to construct to we have to screen to find out our gene of interest. Okay, so let's see. Okay, so our initial idea was to divide the average capacity of average insert capacity of the vector that we are using with the entire genome, right? But that will not giving us the actual number. So under such circumstance, Clarke and Carbon in 1975 gave a formula. This formula looks very easy, that n is equal to ln 1 minus p by ln 1 minus a by b. Okay, so here the n is number of clones that are required or that are to be screened. Okay, if the first one is for the construction of the genomic DNA library and the second one is for the screening of the genomic DNA library, then the p is the probability that the particular DNA fragment will be present. Okay, you will get some percentage like things in your question when the question is based on that formula. So if you get something percent like digit, then make sure they are telling us about p. Okay, and average size of the DNA fragment that can be inserted into the vector. Okay, that means the average DNA size, and the b is total size of the entire genome. Okay, so that is the formula for Clarke and Carbon in 1975. So now we are going to see about a mathematical question that will help you to understand the formula more. Okay, so I want to tell you again the important advice that whenever you will going to solve any kind of mathematical problem, first of all read this question very carefully. Okay, and try to find out what are the tools that they have provided to construct the entire math or to derive the answer. Okay, so let's read the question first. So first of all, lambda umbl3, which is a lambda vector, is used as a vector to clone 20,000 base pair fragments generated from a particular SAU3A digest of the human genome. So here they have provided the total genome size, that means here they have provided the b, right? Similarly, we wish to isolate a gene content completely on a 20,000 base pair fragment. Okay, that means here they are telling us about a, that is average gene size that is inserted to a vector. Okay, so to have a 99% chance, that means it is the value of p, right? Because I have told you the probability of the presence of a particular DNA is p. So I have also told you that you will get the value of p in the form of percentage in most of the time. So this is the p. Okay, now you have to calculate how many independent clones must be examined to find out that 20,000 base pair fragment. Okay, so in the p value we will put 0.99 that I have obtained by dividing 99 with 100. Okay, then in a there will be 20,000 because it is the average DNA size, and in place of b there will be 3 into 10 to the power 9, that is the entire length of the genome. By calculating the equation we will get the number of clones that we have to examine to find out the 20,000 base pair fragment. Okay, so that is how the Clarke and Carbon principle works, and how it is easy to solve any kind of mathematical question coming from genomic DNA library. Okay, so I hope this video will be helpful to you, and thank you for listening to this class. We will again come back with another video where we will going to solve another mathematical problem. Okay, so till then, bye bye friends.